Dear users, welcome to this tutorial which aims to provide a detailed overview on easy navigation and uses of the SCIP database being developed in our lab. So this tutorial will brief you with three key aspects of the SCIP database. The first being the background and the rationale that led to the development of the SCIP database. The second section will deal with the web design and schema that have been used to design and develop the database. And the third part will give you a bird's eye view of the SCIP database, various sections and subsections. So let's first see the first part, the background and the rationale that led to the development of the SCIP database. Combined stress studies in plant being the focus of our lab have led to many valuable in-house publications. We were also interested in collecting and curating various combined stress studies from other labs worldwide. Eventually, we realized that a significant number of the groups are actively publishing in this area, which has led to considerable accumulation of combined stress studies. However, these combined stress studies data is scattered in the literature, which make it really difficult for the researcher to analyze and compare the data sets. So the first step should be to compile, curate and integrate these data into a database so that it can be easily accessible and interpretable by the user. Also, currently, no database or platform solely dedicated to combined stress studies is available in plant. This gap led to the genesis of the SCIP database. It is also important to highlight here that combined stress studies are often complex and are being impacted by various factors like stress interaction, age of the plant, order of the stress, and intensity of the stress. Thus, we integrated several visualization and standalone tools to SCIP database to make it more user friendly and easily accessible to the users. Coming on to the second part of this tutorial, which will brief you with the web design and the schema that have been used to design and develop the database. SCIP database is a relational database and web server. The front end of the database has been rendered using the HTML, which controls the structure of the information displayed to the user. CSS has been used to style the displayed data, while JavaScript and Bootstrap has been used to add interactive web features to the database. At the backend, we have used PHP programming language to code for the web application, while datasets have been stored using the MySQL and XAMPP server. We have also rendered FTP server and standalone BLAS server to the SCIP database. The final part of this tutorial aims to provide the user with a bird's eye view of the various menus and the sub-menus of the SCIP database. After typing the web address, that is www.nipgr.ac.in/cipdb.php, users will be redirected to the home page of the database. This is the home page of the database, which gives access to various menus and sub-menus. So let's first see the various components of the home page. Left section hosts multiple representative images from combined stress studies. Further description can be accessed by hovering over the images. The right section of the home page hosts two important menus, namely the phenomics and the transcriptomics, which can be used to mine morphophysiological and transcriptome combined stress data respectively. Further down, quantitative information of the data points listed in database have been provided. SCIP database currently hosts curated combined stress literature from 734 studies belonging to 122 stress combination covering 325 organisms. Transcriptome studies from 31 literature and 107 trait information have been hosted in the database. To gain further insight of each of these data points, a link to download the corresponding file has been provided. So for example, if the user is interested to know the name of the stress combination being hosted in the SCIP database, on clicking the link, the complete list can be accessed and locally downloaded onto their system. Scrolling further down, a brief introduction about the database along with important literature links have been provided. Definition of important combined stress terminologies have been provided, which can be seen by placing cursor on the highlighted terms. Through the home page, links to other menus like sitemap, author repository, combined stress map, downloads, terminologies, and copyright have also been provided. On clicking the author repository, the user can access the information about various scientists and researchers working in the area of the combined stress worldwide. So currently, this section hosts corresponding author information of 635 different researchers working in the area of combined stress. Through the home page, user can navigate to the About section of the database. So the About section has been further organized into 6 different submenus. So we will be navigating through them one by one. In the FAQ section, multiple frequently asked questions have been hosted for the user to further guide them on various aspects of the SCIP database. On clicking any of these questions, further details can be accessed by the user. The second submenu of About section is the methodology phenome. This section details the method used for phenome data collection, data curation, data interpretation, and final data integration into the SCIP database. It is worthwhile to note that each of these legends is interactive, and on placing the cursor over them, further details about these legends can be accessed. Link to download detailed methodology for phenome have been provided. Also, the list of keywords that we have used to mine phenome dataset, together with list of plant species, organisms, pathogen, and terminologies, can be locally downloaded by the user. The third submenu of the About section is the methodology transcriptome, which details about the methodology used for transcriptome data analysis in the form of the flowchart, together with information about the various visualization hosted in the transcriptome section of the database. User can further download the detailed methodology for transcriptome as PDF using the link provided in the section. The fourth submenu of the About section is the application section. This section of the database aims to demonstrate the users with potential application of the database. It will guide them on how they can use the dataset hosted in the SCIP database to gain several biological insight about combined stress studies. Potential questions of interest and their answers have been hosted under relevant categories. The PDF version of these answers can also be provided and can be locally downloaded by the users. Please refer to the SCIP database application and case study tutorial to gain further detailed insight about the application section of SCIP database. Let's now see the references and links of the section of this database. This section hosts important publication, links, labs, and web resources related to combined stress studies. The left section hosts important publication like reviews, books, and chapters pertaining to combined stress. While in the right section, important web resources, database, general special issues, lab pages, and author publishing in the area of the combined stress have been listed. On clicking the Read More link, the PDF version of these resources can be downloaded together by the users onto their system. This section also provides the user with an option to mine literature information by drop-down based selection. For this, the user needs to choose a category followed by the choice of the subcategory, that is the stress combination of the interest. And on submission, all the references pertaining to that particular stress combination is presented to the user. The references have been further categorized into two sections. The first being the main research articles, for which the corresponding phenome data page is available in our database, and ancillary articles, which are usually reviews, chapter, abstract, thesis, and reports. In case of main research article, two kinds of links have been provided to the user. The first link will redirect the user to either the PubMed or the journal link. While using the second link, view brief content in the data page, the processed phenome data can be assessed by the users corresponding to that particular article. For the ancillary articles, only the link to the PubMed or journal is provided. The final submenu of the About section is the Downloads. This section catalogs and hosts the raw data files, references, and other metadata in the form of the FTP server. From plant breeders' perspective, studies related to genotype characterization under combined stress studies is also provided. The section has been further organized into three subsections. The first section hosts the phenomic datasets. The second section hosts the transcriptome datasets, while in the third section, academic teaching material have been hosted. The phenomic dataset has been further organized based on simultaneous or sequential combined stress category. For each of these categories, three types of files are being provided. The first being the raw data file, the references, and genotype studies if available. On clicking on any of them, the list of the folders with the name of the stress combination is presented to the user. Based on the user interest, user can download the complete raw data for further downstream analysis onto their system locally in the Excel format. Similarly, you can download references and genotype studies. In the second section, all the analyzed data from the transcriptome studies have been hosted. The user can select the plant of interest and download the complete analyzed transcriptome for that particular plant using the link provided. In the third section, multiple teaching materials like links to thesis, posters, protocols, slides, and author repository is provided. All of these can be downloaded locally in the PDF form. A link to completely download the author repository is also provided. This completes the About section of the database. So we will now move to the search section of the database. The search section provides extensive search option for the users to mine the phenomics and the transcriptomics datasets. The section has been further organized into three parts for easy navigation. So let's first see each of these sections one by one. Using the first section, the phenomics datasets can be mined. For this, the user needs to select the particular category, for example, plant, stress combination name, parameter, pathogen, or keyword. So let's see one of them. Suppose the user selects the plant category and enters chickpea. On submission, results will be presented in the form of the interactive bootstrap table. As we scroll down, we can see that 13 hits matching our search is present in the database. The first column of the result provides the serial number followed by the stress combination name, the classification as to whether the study belongs to the sequential or combined stress category, the parameter on the study, the plant name, cultivar information is also provided wherever available, pathogen information is provided, and corresponding reference link is provided, which is either linked to PubMed or journal. Finally, a link to access the phenome data for this article hosted in SCIP database is provided. User can click on this link to go to the corresponding phenomics dataset. All of these results can be downloaded locally by the user using the download result link. The second section can be used to mine the transcriptome datasets. Here again, the first category selection is required. So suppose the user selects stress combination and enters data as nematode and water. On submission, all the differentially expressed genes pertaining to this particular stress combination will be presented in the form of the interactive bootstrap table. So we have 37 entries for this particular search. The table provides information about the gene ID, gene name, stress combination, log fold chain, and pathway and metadata information. Users can interactively visualize the differentially expressed gene links on KEGG pathway wherever possible. So suppose if the user wants to see this gene interactively on KEGG pathway, on clicking the link, the gene in question is highlighted. It is worthwhile to note that on clicking the highlighted gene, several other metadata can be accessed by the users. Using the KEGG genes page, this page also provides links to other important databases like NCBI, TIR, and UniProt. Amino acid sequence and nucleotide sequence information for this gene of interest is also provided. In cases wherever pathway information is not available, differentially expressed genes have been mapped directly to the KEGG genes, and similar information can be accessed for the particular gene. The third section of the search has been designed to accept multiple nucleotide and protein sequences and return potential homologs based on BLASTX and BLAST3 standalone servers. So we will just see an example using the protein sequences. After a sequence input, selection of suitable program is required, and on submission, results is presented in the form of a tabular output, which shows the query, the subject, percentage identity, plant, stress combination name, gene name, and log fold chain. It is worthwhile to note that only the top hit is provided in the form of the table, and these results can be completely downloaded by the user using this link. However, if the user is interested to download complete BLAST result, this link can be used to download the complete BLAST results. Dear users, now we will be moving to the submit section of the database. This section provides users with an option to submit combined stress phenomics or the transcriptomic datasets to the SCIP database. Submission can be done in two ways. Firstly, user can prepare and submit their data using the data submission form. On clicking the link, a Google form is presented. User needs to submit each of these entries, and on submission, data gets automatically submitted. Second option is to prepare your data using the example template provided over here. You can download the blank template file using this link and send your data to the given mail address. Now we will move to the connect section of the database, which further has 4 submenus. The reaches section provides the information and contact information about the corresponding author. In the news and events section, links to important research grant, conferences, and other news is being provided. Links to social networking site are also provided in this section. The second submenu of this connect section is the team member, which provide information about the people involved in this project. In the funding and acknowledgement section, we provide information about the financial support and the acknowledgement to the lab members, scientists, and tools and resources that have been used towards SCIP database design and development. On clicking the view here link, further details about these tools can be accessed. The file and submenu of the connect section is the SCIPDb social. SCIPDb social submenu provides user with an option to connect with us using several social networking sites like Facebook, Twitter, YouTube channel, ResearchGate, and SCIPDb. We have hosted multiple podcasts relevant to combined stress studies in plant. User can listen to these podcasts either on SoundCloud, Spotify, or Google Podcasts. Dear listeners, we will now move to the final section of the database, that is the help section, where we have hosted multiple help and guidance tutorials. The first submenu of the help section is the sitemap, which briefly outlines all the menus and submenus and their content in the database. A link to download the complete PDF of the sitemap is also provided. In the user guide section, users can gain insight with a detailed stepwise tutorial on browsing and mining combined stress data from SCIP database. For example, if a user is interested in mining phenomics dataset, they can click on how to search and mine phenomics dataset. A PDF illustrating stepwise tutorial detailing each step is presented to the user. They can download it locally and use it as a guide to navigate through the SCIP database. Similarly, a second tutorial on how to navigate and understand the visualization of the phenomics data is provided. A tutorial on mining and searching the transcriptome dataset is also provided in detail. Further, how to search and mine metaphenome data is provided. And a tutorial on how to navigate through various menus of SCIP database is also provided. Finally, it is worthwhile to note that SCIP database has a dedicated YouTube channel where we have hosted multiple videos which you can use to gain insight on various aspects of combined stress. Please follow and subscribe to our YouTube channel to get regular updates of new videos being uploaded onto our channel. Thank you for your patient listening.